Sure. Here's a very simple, deeply clear, and detailed summary of the paragraph you gave, written in a way that even someone with no background in science can understand easily. Summary: the beginning of classification of living things, plants and animals. Since ancient times, from the very beginning of human life, people have tried to group or arrange living things, like plants and animals. This is called classification. But in the early days, people did not know science. They grouped living things not with rules or study, but based on how useful those things were to them. For example, they knew cows give milk and meat, trees give wood for houses, sheep give wool for clothes. So they grouped plants and animals based on their use for food, shelter, and clothing. This was done naturally, from instinct, a feeling, not from scientific thinking. Then came Aristotle, a scientist from ancient Greece. After many years, a great thinker named Aristotle, he lived over 2,000 years ago in Greece, tried to make classification more scientific. He studied the outer features, what something looks like of plants and animals. This is called morphology, study of form and structure. How he grouped plants. Aristotle divided plants into 3 groups based on their size and stem type. 1. Trees, big plants with hard, thick trunks. Example, mango tree, neem tree. 2. Shrubs, medium-sized plants with woody branches near the ground. Example, rose, hibiscus. 3. Herbs, small, soft-stemmed plants. Example, grass, mint, spinach. How he grouped animals. Aristotle divided animals into 2 main groups based on whether their blood was red or not. 1. Animals with red blood. Examples, humans, dogs, fish, birds. 2. Animals without red blood. Examples, insects, worms, jellyfish. Why this was important. Aristotle's method was one of the first attempts to classify living things in a scientific way. Even though it was simple and not perfect, it was a big step forward from the earlier way of grouping things only by human use. He tried to look at natural features of living things, like shape, size, and blood, which laid the foundation for modern biology and classification systems. Key points to remember. Idea, explanation, early classification, based on human needs like food and shelter, not science. Instinctive grouping, done naturally, without studying details. Aristotle, first person to use simple science for classification. Plant groups, trees, shrubs, medium, herbs, small. Animal groups. Red-blooded versus non-red-blooded animals. Would you like a visual chart or diagram of this summary to make it even clearer? Or would you like me to explain the next part of the chapter? Sure. Here's a great, deep, and very simple summary of the comic strip that explains the evolution of the classification of living things. Panel 1. Linnaeus's two-kingdom system. In the past, a scientist named Linnaeus created a system to group all living things. He divided them into 2 main groups, kingdoms, plantae, plants, animalia, animals. This system was simple and easy to understand. Example, a tree or grass went into plantae. A lion or human went into animalia. Panel 2. Problems with the two-kingdom system. The 2 groups missed many important differences between living things. The system did not separate eukaryotes, cells with nucleus, and prokaryotes, cells without nucleus. Unicellular, single-celled, and multicellular, many-celled organisms. Photosynthetic, make their own food, like algae, and non-photosynthetic, like fungi. It was like putting very different things in the same box, which caused confusion. Panel 3. Many organisms didn't fit. Some living things didn't clearly belong in plant or animal kingdoms. For example, bacteria euglena or mushrooms didn't fit well in either category. Scientists realized that the 2-group system was not enough. They needed a better way to group life forms. Panel 4. More advanced classification. Over time, scientists learned more about living things, like their cell structure, how they eat, where they live, how they reproduce, and how they evolved. So, new classification systems were made. One of the most accepted new systems is the 5-kingdom system, which includes 1. Monera, all prokaryotes like bacteria. 2. Protista, simple eukaryotes, like amoeba, algae. 3. Fungi, mushrooms, yeast, molds, not plants. 4. Plantae, all true plants. 5. Animalia, all animals. Why this is important. The two-kingdom system was a good start, but it was too simple. Life is very diverse, so a more detailed system was needed. The new systems help scientists understand life better, group organisms more accurately, and study their evolution and relationships. Summary in one line. The two-kingdom system, plants and animals, was simple but too limited, so it was later replaced by more advanced systems, like the 5 kingdoms, which better reflect the real variety and nature of living things. Let me know if you want this made into another comic or further simplified. Here's a deep, simple, and clear summary of the comic strip explaining R.H. Whittaker classification, as if teaching a total beginner or a child. 1. Whittaker. Only 2 kingdoms. In the beginning, scientists used a very simple system. 2 groups. Plantae. Plants. Animalia. All put into just these 2 boxes. If it didn't move and made its own food, it was called a plant. If it moved and ate food, it was called an animal. Problem. Many living things didn't fit properly in either group. For example, bacteria, tiny, no nucleus. Fungi, like mushrooms. Algae, some look like plants, some don't. These didn't really belong fully to plant or animal. 2. Whittaker's new idea, 1969, 5 kingdoms. Scientist R.H. Whittaker said, let's not just look at what they look like. Let's also see what their cells are like, how they eat, and how they reproduce. He made a better, deeper system. 5 kingdoms, not just 2. These 5 are 1. Monera, tiny simple organisms with no nucleus, like bacteria. 2. Protista, small simple organisms with a nucleus, like amoeba, algae. 3. Fungi, like mushrooms which absorb food but don't photosynthesize. Plantae, all plants that make food by photosynthesis. 5. Animalia, all animals that eat other organisms and move. 3. What did Whittaker use to classify? Main criteria. Whittaker looked at 5 main things to decide what group something belongs to. What it means. 1. Cell structure. Does it have a nucleus or not? 2. Body organization. Is it just one cell or made of many cells? Are cells organized into tissues? 3. Mode of nutrition. Does it make food by photosynthesis, absorb food, or eat other things? 4. Reproduction. How does it reproduce? Asexually or sexually? 5. Evolutionary relation. How closely is it related to others in terms of evolution? 4. Later, the 3-domain system. Scientists later broke Monera into 2 parts because bacteria actually come in 2 very different types. 1. Eubacteria, normal bacteria. 2. Archaebacteria, ancient bacteria, live in extreme places. This led to 3 domains. Domain 1, bacteria. Domain 2, archaea. Domain 3, eukarya, everything with a nucleus, protists, fungi, plants, animals. So this newer system leads to 6 kingdoms, but you will learn that in higher classes. Summary in simple words. Before, only 2 groups, plants and animals. Whittaker, 1969, said, that's too simple. Let's use 5 groups based on cell type, number of cells, how they eat, how they reproduce, evolution history. So he made 5 kingdoms: Monera, bacteria; Protista, single-celled eukaryotes; fungi, mushrooms and mold; Plantae, all plants; Animalia, all animals. Let me know if you'd like a separate comic for the 3-domain system or a side-by-side chart of all 5 kingdoms explained visually. Great. Let's now go even deeper, explaining each line and word of the 5 kingdoms table with super simple, clear, and deep explanations, like I'm teaching a total beginner or a child who has no science background at all. What is the 5-kingdom classification? Scientists found that all living things are not the same. So they grouped all living things into 5 big categories. These categories are called kingdoms. Each kingdom is like a giant family of living things that share similar features. The 5 kingdoms are 1. Monera, smallest, simplest life, like bacteria. 2. Protista, simple but more developed, like algae. 3. Fungi, organisms like mushrooms and yeast. 4. Plantae, all types of plants. 5. Animalia, all animals, including humans. Now, let's understand the key features scientists use to divide these groups. Table 2.1, characteristics of the 5 kingdoms. Let's go line by line, word by word. 1. Cell type. This means, what kind of cell is the organism made of? Every living thing is made of cells, but not all cells are the same. There are 2 main types of cells. A. Prokaryotic. Pro means before. Karyon means nucleus, the brain control center of a cell. So prokaryotic equals before nucleus. These cells have no true nucleus. The DNA is floating loose in the cell. These cells are very tiny and simple. Example, bacteria had a B. Eukaryotic. U means true or real. Eukaryotic equals true nucleus. These cells have a nucleus with a covering. DNA is safely stored in the nucleus. The cell has many parts, organelles, doing special jobs. Examples, amoeba, plants, animals. Table for cell type kingdom. Cell type, meaning example, more complex. Plants, Monera, prokaryotic, very simple, no true nucleus. Bacteria, Protista, eukaryotic, has real nucleus. Amoeba euglena, fungi, eukaryotic, real nucleus, more complex. Mushroom, yeast. Plants, eukaryotic, advanced cells with nucleus. Rose mango tree. Animals, eukaryotic, complex cells with true nucleus. Humans, dogs, fish. Nashrantar 2. Cell wall. This means, does the cell have a hard outer wall to protect it? Think of the cell wall like a strong fence around a cell. Some cells have it, like a box. Some do not, like a soft balloon. Let's break this down. Kingdom. Present, made of, notes. Monera. Yes. Not cellulose, but special polysaccharides plus amino acids. Cell wall helps bacteria survive in harsh places. Protista, sometimes, varies. Some like algae have it, others like amoeba don't. Fung. Yes. Chitin, same material as insect shells, very strong. Yes. Cellulose, makes plants firm, also in veggies, paper. Animals, no. Only have a cell membrane, soft skin. Extra info, polysaccharide, long chain of sugars. Chitin, tough material found in mushrooms and insects. Cellulose equals tough plant fiber in leaves, vegetables. 3. Nuclear membrane. This means, does the nucleus cell's brain have a cover around it? Think of a nuclear membrane like a wall around a boss's office. It protects the DNA, the instruction manual of life. Kingdom, nuclear membrane, present? Explanation. Monera. No. DNA floats freely, no separate nucleus. Protista? Yes. DNA is inside a nucleus with a wall. Fungi, yes. Well-protected nucleus. Plants, yes. Same as animals. Animals, yes. DNA safely inside nucleus. 4. Body organization. This means, how is the whole body built? Are they made of one cell or many? Do the cells form tissues or organs? Let's break down some important words. Unicellular, made of just one cell, like a single-room house. Multicellular, made of many cells, like a big building. Tissue equals a group of similar cells doing one job, like bricks in a wall. Organ is a part of the body doing a special job, like the heart or leaf. Organ system, many organs working together, like digestive system. Table, body organization. Kingdom, body organization. Example, Euglena, Monera, unicellular, one tiny cell, bacteria. Protista, unicellular, one cell that acts like a tiny animal or plant. Amoeba. Fungi, multicellular or loose cell groups. Mushrooms have many cells. Plants, tissues and organs. Roots, leaves, stems, all are organs. Animals, organs and organ systems. Heart, brain, stomach, all work together. Number 5. Mode of nutrition. This means, how do they get food? There are 2 basic types. 1, autotrophic. Auto means self. Trophic means feeding. They make their own food, like a chef cooking for themselves. Example, plants use sunlight to make food, photosynthesis. 2. Heterotrophic. Hetero means other. They depend on others for food, like ordering food. Types: saprophytic, eat dead or decaying things. Mushrooms. Parasitic, live on or inside another living thing and steal food, like tapeworm. Holozoic, eat solid food and digest it inside, like humans, lions. Table, mode of nutrition, type of nutrition, explanation example. Algae make food. Monera, both autotrophic, photosynthesis or chemosynthesis, and heterotrophic. Some bacteria make food, others eat from surroundings. Protista, both autotrophic and heterotrophic. Algae make food, amoeba eats things. Fungi, heterotrophic, saprophytic, parasitic. Mushroom grows on dead things. Plants, autotrophic photosynthesis. Plants use sunlight, water, CO2. Animals, heterotrophic, holozoic. Humans, lions eat food and digest it. Final easy recap. Feature: Monera, Protista, fungi. Plants, animals, sunlight, no. Cell type: prokaryotic, eukaryotic. Eukaryotic, eukaryotic, eukaryotic. Cell wall: yes, sometimes yes, yes, no. Nucleus cover: no, yes. Yes, yes, bond cell, one cell, many cells. Organs, organ systems. Nutrition: mixed, eats dead others, makes own food, eats others. Real-life examples for each kingdom. Kingdom, real-life example. Monera, bacteria on your skin or in yogurt. Protista, amoeba in dirty water, algae in ponds. Fungi, mushrooms, mold on bread. Plants, rose grass, coconut tree. Animals, human, cat, bird, earthworm. Would you like a colorful diagram? A quiz to check your understanding? Flashcards or memory tricks? I'm happy to help you master this in any way you like. Just tell me. Sure. Here's a great, deep, and very simple summary of everything you just learned, written like a total beginner's guide, with clear language and examples to help you understand everything fully. Summary: why the old plant grouping system didn't work, and what it taught us. What was the old system? Before scientists created the Five Kingdoms system, they used a very simple way to classify living things. They said, if it has a cell wall, it's a plant. So they put the following into the plant kingdom, included in plants. Examples: bacteria, tiny single-celled organisms, blue-green algae, found in water, now called cyanobacteria. Fungi, mushrooms, mold, yeast, mosses. Small green plants in damp places, ferns. Green leafy plants without flowers, gymnosperms. Pine trees, no flowers but seeds, angiosperms. Flowering plants like roses or mango trees. Why did they group them all as plants? Because they all had cell walls, a hard covering around their cells. That was the main reason. But this was a problem. Why? Let's go point by point and understand. Problem 1. Mixed, simple, and complex organisms. Bacteria and blue-green algae are prokaryotes. Very simple cells, no nucleus. Real plants like mosses, ferns, and trees are eukaryotes, complex cells with nucleus. They look very different inside, but were still placed together just because of cell walls. Example: like putting a toy phone and a real smartphone in the same box just because both have buttons. That's confusing. Problem 2. Mixed unicellular and multicellular organisms. Some plants had only one cell, like Chlamydomonas. Others had many cells, like Spirogyra or ferns, but the old system grouped them together as algae. Example: like saying a single brick and a whole building are the same thing. Not accurate. Problem 3. Mixed autotrophs and heterotrophs. Autotrophs make their own food using sunlight, photosynthesis. Example: green plants. Heterotrophs cannot make food. They absorb or eat it. Example: fungi. Fungi don't do photosynthesis at all, yet were put in the same group as green plants. Example: like putting a cook and a baby in the same group just because both are in the kitchen. Absolutely. Let's now take the entire paragraph and explain it even more deeply, step by step, in the simplest words possible, like you're learning for the first time. I will break every sentence down, add examples, and give extra detail wherever needed so that nothing feels confusing. Original text. When such characteristics were considered, the fungi were placed in a separate kingdom, Kingdom Fungi. Super simple explanation. Let's go slowly. Scientists looked at certain features of organisms. These features are things like how they eat, what their cells look like, if they can make their own food, photosynthesis, what their cell wall is made of. They found that fungi, like mushrooms, molds, and yeast, are not like plants. Example, plants make food using sunlight, photosynthesis, but fungi don't. Fungi get food by absorbing it from dead or decaying things. Also, their cell wall is different. Plants have cellulose in their walls. Fungi have chitin in their walls. Because of these differences, fungi were moved out of the plant kingdom and given their own kingdom called Kingdom Fungi. Original text. All prokaryotic organisms were grouped together under Kingdom Monera. Super simple explanation. What does prokaryotic mean? These are very, very tiny living things. Their cells are very simple. They do not have a nucleus inside the cell. These include bacteria, blue-green algae, also called cyanobacteria. So, all of these very simple one-celled organisms were put together into one group called Kingdom Monera. Example, the bacteria in curd, yogurt, the bacteria that cause sickness, the blue-green algae in water. All of these belong to Kingdom Monera, original text, and the unicellular eukaryotic organisms were placed in Kingdom Protista. Super simple explanation. What does unicellular eukaryotic mean? Unicellular equals made of only one cell. Eukaryotic equals their cell has a nucleus, a brain-like part inside the cell. So these are tiny, single-celled organisms that are more advanced than bacteria. They were put into a group called Kingdom Protista. Original text. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants and both having cell walls. Super simple explanation. Earlier, green single-celled organisms like Chlamydomonas, chlorella were placed in the plant kingdom because they were green. They did photosynthesis, made their own food. They had cell walls. But they are also single-celled and have a nucleus. So now, scientists said, they don't really belong with big plants like mango trees and grass. So they moved Chlamydomonas and chlorella to Kingdom Protista. Original text. With Paramoecium and amoeba, which were earlier placed in the animal kingdom which lack cell wall. Super simple explanation. Before, Paramecia and amoeba were thought to be part of the animal kingdom because they move, they eat food from outside, they don't have a cell wall, just like animals. But now scientists said, wait, these are also single-celled with a nucleus. So they also moved Paramecia and amoeba into Kingdom Protista. Original text. It has put together organisms which in earlier classifications were placed in different kingdoms. Super simple explanation. So now, Kingdom Protista has organisms that were earlier called plants, like Chlamydomonas, and organisms that were earlier called animals, like amoeba. They all are now together, not because they look the same, but because they are all single-celled, eukaryotic, have nucleus. Original text. This happened because the criteria for classification changed. Super simple explanation. Why did this happen? Because scientists changed the way they group living things. Before, they grouped based on outside appearance. Now, they group based on what's inside the cell, how they get food, how they reproduce, and how they are evolutionarily related. Original text. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Super simple explanation. Science is always growing. So in the future, the classification may change again if we find new information. We better understand evolution, which organisms came from which. As our knowledge grows, so will our grouping systems. Original text. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, i.e., is based on evolutionary relationships. Super simple explanation. Scientists now try to group organisms in a way that shows 1. Morphology, how they look. 2. Physiology, how their body works. 3. Reproduction, how they make babies. 4. Phylogeny, how they are related by evolution, family tree. Now we care about how living things are connected by history. Who came from whom? Original text. In this chapter, we will study characteristics of kingdoms Monera, Protista, and fungi of the Whittaker system of classification. Super simple explanation. In this chapter, you will learn about Monera, very simple, no nucleus, like bacteria. Protista, single-celled, has nucleus, like amoeba, paramecium. Fungi, absorb food, cell wall of chitin, like mushrooms and mold. This system was created by a scientist named R.H. Whittaker. Original text. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapters 3 and 4. Super simple explanation. Later, in chapter 3 and chapter 4, you will learn in detail about Plantae equals all the green plants. Animalia, all the animals, including humans, insects, dogs, fish, etc. Full super simple summary. Kingdom, cell type, examples, special features. Monera, prokaryotic, no nucleus. Bacteria, blue-green algae, very simple, tiny, one-celled. Eukaryotic, has nucleus. Amoeba, paramecium, chlamydomonas, one-celled, can be plant-like or animal-like. Fungi, eukaryotic, mushroom, mold, yeast, absorb food, chitin cell wall. Plantae, eukaryotic, trees, grass, algae, make food by sunlight, cellulose wall. Animalia, eukaryotic, humans, fish, insects. Eat food, can move, no cell wall. Would you like me to now explain Kingdom Monera in this same deeply simple style, one line at a time? Absolutely. Let me now explain the entire passage again, but this time in even greater detail, in the simplest, easiest way possible. Like I am teaching a total beginner, even a child. I'll go word by word, line by line, and I will add more examples, extra information, and full clarity wherever needed. Add line 1. Kingdom Monera. Let's break this down. Kingdom. In biology, we divide all living things into big groups. These big groups are called kingdoms. It's like sorting all living things into families. Monera. This is the name of one of those big groups. It is the group that contains bacteria. So, Kingdom Monera means the group of living things that includes bacteria. All bacteria belong to this group. Line 2. Bacteria are the sole members of the Kingdom Monera. Let's explain every word. Bacteria. These are tiny living things, so small that you can't see them with your eyes. You need a microscope. Are the sole members. This means they are the only ones in this group. Sole equals only. Of the Kingdom Monera. They are the only living things in this Monera group. So, only bacteria belong to Kingdom Monera. There are no other living things in this kingdom. Line 3. They are the most abundant microorganisms. Let's explain. They, this means bacteria. Most abundant, this means they are found in the largest numbers. There are more bacteria than any other kind of tiny living thing. Microorganisms. These are living things that are so small, you need a microscope to see them. Micro equals tiny. Organisms, living beings. So, bacteria are everywhere and very, very common. There are more bacteria than any other small living thing on Earth. Line 4. Bacteria occur almost everywhere. Word by word. Bacteria, the tiny living things. Occur. This means they exist or they are found. Almost everywhere. This means in nearly every place on Earth. So, bacteria live in nearly every place you can think of. In water, soil, air, and even inside our bodies. Line 5. Hundreds of bacteria are present in a handful of soil. Word by word. Hundreds of bacteria. Tellidae, a very large number of bacteria. Even just one handful has many. Are present. They exist or are inside. In a handful of soil, just a small amount of dirt from the ground. So, even if you take a small handful of soil, there are hundreds and hundreds of bacteria living in it. That shows how common they are. Line 6. They also live in extreme habitats, such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Word by word, they, bacteria, also live, they can survive. In extreme habitats, this means in very hard, dangerous, or unusual places, such as hot springs, deserts, snow, and deep oceans. These are examples of extreme places. Hot springs, very, very hot water. Deserts equals very dry and hot. Snow was freezing cold. Deep oceans, dark, cold, high pressure. Live in places where almost nothing else can survive, like boiling hot water, freezing snow, or deep under the sea. Line 7. Many of them live in or on other organisms as parasites. Word by word, many of them, many bacteria, live in or on. They stay inside or on the surface of other living things. Other organisms, other living beings like humans, animals, or plants. As parasites. A parasite is a living thing that lives on another being and takes its food or energy, often harming it. So, many bacteria live inside humans or animals, and some of them cause sickness because they are parasites. Example, bacteria that cause stomach pain or sore throat live inside us as parasites. Line 8. Bacteria are grouped under 4 categories based on their shape. Word by word. Grouped, they are divided into types. 4 categories. There are 4 different types. Based on their shape, the difference is in their shape or form. So, bacteria come in 4 different shapes, and scientists use these shapes to put them into groups. Line 9. The spherical coccus, cocci, word by word. Spherical, round like a ball or circle. Coccus, this is the name of round-shaped bacteria. Cocci, pill, means plural. One is coccus, many are cocci. So some bacteria are round. These are called coccus 1 or cocci many. Example, streptococcus is a cocci-shaped bacteria that causes sore throats. Line 10. The rod-shaped bacilli, rod-shaped, like a stick or long rectangle. Bacillus, name for rod-shaped bacteria. Bacilli, plural form is bacillus. So, some bacteria look like rods or sticks. These are called bacillus 1 or bacilli many. Example, E. coli is a rod-shaped bacillus bacteria found in our intestines. Line 11. The comma-shaped vibrio. Comma-shaped, like a curved line, like the comma punctuation mark. Vibrium, name for this type. Vibrio, plural is vibrio. So, some bacteria look like a comma, comma. These are called vibrio. Example, Vibrio cholerae causes cholera, a disease from dirty water. Line 12. And the spiral spirillum, spirilla. Spiral, like a coil, twist, or curly spring. Spirillum, name for this spiral-shaped bacteria. Spirilla, plural form. So, some bacteria look like spirals or corkscrews. They are called spirillum 1, spirilla many. Example, Spirillum minus can infect wounds from animal bites. Line 13. Though the bacterial structure is very simple, they are very complex in behavior. Word by word, though, even if structure is very simple, bacteria have a very basic body, no complex organs or parts. They are very complex in behavior. But the way they act or work is very smart and tricky. So even though bacteria have a simple body, they can do very complicated things, like making food, moving, causing diseases, or living in strange places. Line 14. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Let's break this down. Compared to many other organisms, when you look at bacteria versus other living things, bacteria as a group, all types of bacteria together, show the most extensive, show the greatest, widest range. Metabolic diversity. This means they have many different ways of getting and using energy, metabolism, how the body works to stay alive. So, bacteria can live in many different ways. Some make their own food, some eat others, some breathe oxygen, some don't. This makes them very diverse and unique. Line 15. Some of the bacteria are autotrophic, i.e. they synthesize their own food from inorganic substrates. Word by word, some are autotrophic. Some bacteria make their own food, i.e. they synthesize their own food. Synthesize means to build or make. From inorganic substrates, they use non-living materials like sunlight, carbon dioxide, or chemicals to make food. So, some bacteria can make food by themselves using things like sunlight or chemicals, just like plants do. Line 16. They may be photosynthetic autotrophic or chemosynthetic autotrophic. Word by word, photosynthetic autotrophic. These bacteria use sunlight to make food, like plants. Example, cyanobacteria, blue-green algae. Chemosynthetic autotrophic. These bacteria use chemicals, not sunlight, to make food, like those in deep-sea vents. So, some bacteria use sunlight, others use chemicals to make their own food. Line 17. The vast majority of bacteria are heterotrophs, i.e., they depend on other organisms or on dead organic matter for food. Word by word, the vast majority, most bacteria, are heterotrophs. They cannot make their own food. They get it from outside, i.e., they depend on other organisms. They eat or live on other living things, or on dead organic matter, or they eat dead plants, animals, or waste. So, most bacteria get food by eating other things, either living things or dead stuff. Example, bacteria that help rot dead plants or cause infections. Final summary, simple points. 1. Monera is the kingdom of bacteria only. 2. Bacteria are tiny, everywhere, and very common. 3. They live even in very harsh places, like deserts or hot springs. 4. Some live inside animals, sometimes causing disease. 5. Bacteria come in 4 shapes: round, coccus, rod, bacillus, comma, vibrio, spiral, spirillum. 6. Bacteria are simple in body, but very smart in how they live. 7. Some bacteria make their own food, with sunlight, photosynthetic, with chemicals, chemosynthetic. 8. But most bacteria eat other living or dead things. Would you like me to turn this into flashcards, quiz questions, or pictures to help you remember it better? Here is a deep, clear, and simple summary of the entire explanation about archaebacteria, written like you're learning from the world's best teacher in the easiest possible way, with all the key ideas and examples. Summary of archaebacteria. The special survivors. What are archaebacteria? Archaebacteria are a very old and special group of bacteria. They are microscopic, too tiny to see without a microscope. They are different from normal bacteria because they can live in extreme and harsh environments where most other living things cannot survive. Why are they special? They live in places that are too salty, too hot, too acidic, or have no oxygen. Their cell wall, outer covering, is built differently. This makes them strong and tough. This special wall helps them survive in environments where most life would die. Types of archaebacteria, 3 main kinds. 1. Halophiles, salt lovers, live in very salty water, like in salt lakes or the Dead Sea. The word comes from halo equals salt, phile equals lover. Example, bacteria in salt ponds or salt mines. 2. Thermoacidophiles, heat and acid lovers, live in very hot and acidic places, like hot springs or near volcanoes. The word comes from thermo, heat, acido, acid, phile, lover. Example, bacteria in Yellowstone hot springs, where water is boiling and sour. 3. Methanogens, methane makers, live in places with no oxygen, like muddy swamps or the guts of animals like cows and buffaloes. They produce methane gas, also called biogas. The word comes from methano, methane, gen, maker. Where are methanogens found? Inside the stomachs, guts of ruminant animals like cows, buffaloes, goats, and deer. These animals eat grass, and methanogens help digest the food. The methanogens release methane gas during digestion. This is why cow dung can be used to make biogas. Real-life use. Biogas from cow dung. In many villages, people collect cow dung and put it into a biogas tank. Methanogen bacteria in the dung eat the waste and release methane gas. This gas is used for cooking, lighting, and fuel. It's a clean energy source. Key features of archaebacteria. Feature, what it means, very old, one of the earliest life forms on Earth. Tiny. can only be seen with a microscope. Survive extreme conditions. Salt, heat, acid, no oxygen. They're super tough. Special cell wall, strong, different from other bacteria, protects them. Make biogas, especially methanogens, used as fuel in homes. Why should we care? They help us understand how life can survive in extreme places, even on other planets. They are used in science, energy, and maybe even in space research. They help reduce pollution by turning waste like dung into useful energy. Super simple final takeaway. Archaebacteria are ancient bacteria that live in harsh places like salty lakes, hot springs, and animal guts. They are different from normal bacteria because of their strong outer wall. Some of them, called methanogens, help make biogas from dung, which is used for cooking and fuel in many homes. Would you like a visual diagram or a flowchart next to remember this better? Here is a great, deeply detailed, and very simple summary of the full explanation on eubacteria, especially the kinds like cyanobacteria and chemosynthetic bacteria, written step by step like teaching a beginner or child. Summary of eubacteria, true bacteria in deep detail and simple language. 1. What are eubacteria? Eubacteria are also called true bacteria. They are found everywhere in water, soil, food, air, inside your body, even in extreme places like hot springs. There are thousands of types, and more are still being discovered. They are the most common and best known type of bacteria. 2. Body features of eubacteria. They have a rigid cell wall. This wall gives them shape and protects them like armor. Some can move. If they do, they use a flagellum. A flagellum is a tiny tail that spins like a propeller to help them swim. Visual example. Imagine a hard jelly bean with a tail. That's what a motile eubacterium looks like. 3. Cyanobacteria, special plant-like bacteria. Cyanobacteria are a type of eubacteria that can do photosynthesis, make food using sunlight. They are also called blue-green algae, but they are not algae, they are bacteria. They have chlorophyll A, the same green pigment found in plants. So, they use sunlight, carbon dioxide, and water to make their own food, just like plants. Think of them as tiny green chefs that cook using sunlight. 4. Shapes and habitats of cyanobacteria. Shape. They can live as unicellular, single cell, colonial, groups of cells sticking together, filamentous, long threads of cells. Habitats. They can live in freshwater, ponds, lakes, marine water, oceans, terrestrial places, on soil, rocks, tree bark. Many look like green slime or threads in water. 5. Protective coating, gelatinous sheath. Cyanobacteria colonies are often surrounded by a jelly-like covering called a gelatinous sheath. This protects them and keeps the colony together. Picture peas trapped inside jelly. That's a bacterial colony in a gelatinous sheath. 6. Blooms in polluted water. In dirty water with lots of nutrients, like sewage or fertilizer runoff, cyanobacteria can multiply rapidly. This creates blooms, thick layers of bacteria floating on the water surface. These blooms can be harmful. They block sunlight, use up oxygen, and kill fish and plants. Example, green scum or slime covering a pond after rain. 7. Nitrogen fixation, a superpower. Some cyanobacteria can take nitrogen from the air and convert it into a form that plants can use. This is called nitrogen fixation. It happens in special cells called heterocysts. Heterocysts have thick walls and only do the job of nitrogen fixation. Bacteria like nostoc and anabaena live with plants and naturally fertilize the soil. 8. Chemosynthetic autotrophic bacteria, chemical energy makers. These bacteria do not need sunlight. Instead, they get energy by breaking down chemicals like ammonia, nitrites, nitrates. When they do this, they release energy, which they use to make ATP, the energy molecule used in cells. They are called chemosynthetic autotrophs because they make their own food using chemicals. Imagine little machines in the soil that break down waste chemicals and make energy. 9. Recycling role in nature. These bacteria help in recycling important nutrients like nitrogen for leaves, phosphorus for roots and fruits, iron for energy processes, sulfur for building proteins. They break down dead organisms and waste, returning to the soil and water. Without them, plants couldn't grow, and nature's food chain would break. Final deep simple summary. Feature. What it had wall plus flagellum, and nature's food protection, some eubacteria, true bacteria found everywhere, in water, soil, air, and even in you. Cell wall plus flagellum, hard wall for protection. Some use a tail to swim. Cyanobacteria, plant-like bacteria that make food using sunlight, unicellular, colonial, filamentous, single cells, clusters, or long threads. Habitats, freshwater, saltwater, or land. Gelatinous sheath, jelly-like coating that protects colonies. Blooms, rapid overgrowth in polluted water, forming green scum. Heterocysts, special cells that convert air nitrogen into soil nutrients. Chemosynthetic bacteria, make energy from breaking down chemicals, not sunlight. Nutrient recycling, they return nitrogen, phosphorus, iron, and sulfur to nature. Let me know if you'd like a labeled diagram, a short memory trick, a practice quiz, or a comparison chart between cyanobacteria and chemosynthetic bacteria. I'm happy to help further. Sure. Here's a great, deep, and very detailed summary of the full explanation above, written in the simplest language possible, like teaching a total beginner. This summary will cover all the key ideas and add helpful examples so you fully understand. Detailed summary. Heterotrophic bacteria and mycoplasma. 1. Heterotrophic bacteria. What are they? These bacteria cannot make their own food. They get their food by eating or absorbing nutrients from other living things, like plants and animals, or dead materials, like dead leaves, waste, etc. They are called heterotrophic because hetero equals other, trophic equals feeding. Key point. These bacteria are like humans. We can't make our own food like plants do, so we also depend on outside sources. Most common type of bacteria. These heterotrophic bacteria are the most abundant, most in number in the natural world. They are found in soil, water, air, inside plants and animals, even inside the human body. Key point, they are everywhere in nature, more common than any other type of bacteria. 3. Most are decomposers, cleaners of nature. A large number of these bacteria are decomposers. They break down dead plants, dead animals, waste materials. They convert these things into simple nutrients, which go back into the soil. Why is this important? Without them, dead things would pile up and nutrients would be lost. They keep the environment clean and help plants grow better. 4. Some are very useful to humans. Many heterotrophic bacteria are beneficial, helpful in our daily lives. Making curd from milk. Bacteria like lactobacillus convert milk into curd. They do this by turning the milk sugar, lactose, into lactic acid, which makes the milk thick and sour. B. Producing antibiotics. Some bacteria, like streptomyces, make antibiotics, medicines that kill harmful bacteria. These antibiotics help us recover from infections. C. Nitrogen fixation in plants. Bacteria like rhizobium live in the roots of legume plants, peas, beans, etc. They take nitrogen gas from the air and convert it into plant food. This helps plants grow without needing fertilizers. Key point. These bacteria help in food, farming, and medicine. 5. Some are harmful pathogens. Not all bacteria are good. Some are pathogenic. This means they cause diseases. These harmful bacteria can infect humans, e.g., cause fevers, diarrhea. Crops, e.g., destroy fruits or leaves. Farm animals, e.g., cause illness in cows. Pets, e.g., infect dogs or cats. Examples of diseases caused by bacteria. Cholera causes severe diarrhea, spread through dirty water. Typhoid affects the stomach, also spreads through unsafe water. Tetanus causes painful muscle stiffness, enters through dirty wounds. Citrus canker damages citrus fruits like lemons and oranges. #6. How do bacteria reproduce, multiply? A. Binary fission, main method. One bacterium splits into two. Each new bacterium is a copy of the original. This happens very quickly. Under good conditions, a bacterium can double every 20 minutes. B. Spores in tough times. When food or water is not available, bad conditions, bacteria make spores. Spores have thick coverings that protect them until things improve. When conditions are good again, the spores become active and grow. C. DNA sharing, primitive sex-like reproduction. Sometimes bacteria can transfer DNA from one to another. This is not true sexual reproduction, but a simple way of sharing genes. It helps them adapt to new environments or resist antibiotics. 7. Mycoplasma. Special bacteria-like creatures. No cell wall. Mycoplasma are tiny organisms that do not have a cell wall. Most bacteria have a strong wall, but mycoplasma do not. This makes them flexible and hard to kill. B. Smallest living cells. They are the tiniest living cells ever discovered. Some are even smaller than regular bacteria. C. Can live without oxygen. They are anaerobic, meaning they can survive without oxygen. Some are harmful. Many mycoplasma are pathogenic. They cause diseases in plants and animals. Example, mycoplasma can cause pneumonia in humans. They can also infect crops, making them yellow or weak. Super short final summary. Heterotrophic bacteria eat from outside sources and are very common in nature. They clean the environment, help make food and medicine, and help plants grow. Some bacteria are harmful and cause diseases in humans, animals, and crops. They mostly reproduce by splitting in two and can form spores or share DNA. Mycoplasma are tiny, wall-less, oxygen-free living organisms. Some are dangerous pathogens. Would you like a chart, mind map, or revision flashcards for this content too? Deep, detailed summary of Kingdom Protista in very simple, clear language. 1. What is Protista? 1. One-celled eukaryotes. Protists are living things made of only one cell, single-celled. Their cell is a eukaryotic cell, meaning it has a true nucleus, a distinct control center inside the cell, and other membrane-bound organelles, little machines that do specific jobs. 2. Why Protista? Scientists grouped all these single-celled eukaryotes under the name Protista because they don't fit neatly into the plant, animal, or fungus kingdoms. Protists are a kind of bridge or in-between group. Some behave like plants, some like animals, some like fungi, and some share features of more than one. 3. Classification confusion. The boundaries of Protista are not clear-cut. Example, one scientist may call a green protist that makes food from sunlight a plant, while another calls it a protist. Because of these overlaps, Protista remains a mixed category where scientists sometimes disagree on what belongs. Nashar 2. Major types of protists, 5 groups. In this summary, we will cover the 5 main groups included in Protista: chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans, with simple examples. 2.1 Chrysophytes, golden algae. Where they live, mostly in freshwater ponds and lakes. Color and food, often golden brown or yellow-green, because they have special pigments for photosynthesis, making their own food from sunlight. Shape and shell. Many have a hard shell or cell wall made of glass-like silica, forming beautiful symmetrical patterns. Example, diatoms are a type of chrysophyte. Each diatom cell has a glass box cover with intricate designs. They float in water and produce oxygen when they photosynthesize. Role in nature, make a lot of the oxygen in lakes and seas. Serve as a primary food source, tiny plants in the water that small animals and fish eat. 2.2 Dinoflagellates. Where they live, mostly in saltwater, ocean seas, but some in freshwater. Shape and movement. Each has 2 flagella, whip-like tails. One wraps around the middle, the other trails behind. These tails let it spin and move through water. Food and light. Many are photosynthetic, contain chlorophyll. They look greenish or reddish. Some are mixotrophic. They both photosynthesize and eat other tiny creatures. Special features. Some glow in the dark, called bioluminescence. Certain dinoflagellates in large numbers cause red tides. The water turns red and can be toxic to fish. Example, goniolax often glows at night in warm seas. Role in nature, part of phytoplankton, tiny floating plants that form the bottom of the food chain in oceans. Can be both helpful, oxygen producers, and harmful, red tide toxins. Shaknar 2.3 euglenoids, e.g. euglena. Where they live, freshwater, ponds, slow streams. Shape and movement. They have one flagellum, a long tail, that they whip back and forth to swim. Their cell wall is flexible. No rigid cell wall like plants. They can change shape. Food. In sunlight, they use chloroplasts to do photosynthesis like a plant. They turn sunlight into food and appear green. In darkness, no sunlight, they behave like animals. They move around and eat tiny particles or bacteria. Example, Euglena gracilis, green, slim, swims around and has an eye spot that helps it find light. Role in nature. Provide oxygen through photosynthesis. Serve as food for small aquatic animals. Show a clear bridge between plants and animals because they can do both. 2.4 slime molds, where they live. Moist, damp places, on rotting logs, wet soil, leaf litter. Appearance and movement. When alone, each cell is tiny and looks like slime. In search of food, thousands of cells can come together to form a big, blob-like mass that moves very slowly over surfaces, called a plasmodium. Food. They feed on bacteria and decaying plant material. As they move, they eat by engulfing food particles. Reproduction. When food becomes scarce, they form a fruiting body, a stalk with a spore-filled top. Spores release into the air, land in damp spots, and grow new slime molds. Example, Physarum polycephalum, a bright yellow slime mold you might see on a damp log. Role in nature. Recycle nutrients by breaking down dead matter. Serve as food for tiny insects and other organisms. 2.5 protozoans, tiny animal-like protists. Where they live, freshwater, marine, sea, or inside other animals. Movement and feeding. Many move and feed like tiny animals. Amoeba crawls by extending false feet, pseudopodia, and surrounds its food. Paramecium uses cilia, short hair-like projections, all over its surface to swim and sweep food into a mouth-like area. Plasmodium, the malaria parasite, lives inside red blood cells of humans and mosquitoes. Reproduction. Many reproduce asexually by simply splitting in two, binary fission. Some have complex sexual cycles, mixing DNA from two cells to form a new cell. Examples, Amoeba proteus, shapeless, constantly changing form, found in pond water. Paramecium caudatum, slipper-shaped, swims smoothly using cilia. Plasmodium falciparum, causes malaria in humans. Role in nature and health. Many protozoans are beneficial. They help keep bacterial populations in check and serve as food for small creatures. Some are harmful. They cause diseases, e.g. malaria, sleeping sickness. Number 3. Where do protists live? Mostly aquatic, freshwater, ponds, lakes, rivers, amoeba, euglena, many diatoms. Marine, saltwater, oceans, seas, dinoflagellates, some diatoms. Damp soil, decaying logs, leaf litter, especially for slime molds and certain protozoans. Why water? Protists need a wet environment because they absorb nutrients and oxygen directly through their cell membrane, no hard skin. They often move by swimming or gliding, so they need fluid around them. Many depend on water currents to bring food, like bacteria or tiny plant particles. Number 4, protist cell structure. 1. Nucleus, a central control center that holds DNA, the instructions to run the cell, visible under a microscope as a dark spot inside the cell. 2. Membrane-bound organelles, the little machines inside. Mitochondria, powerhouses, produce energy from food. Chloroplasts in photosynthetic protists contain green pigment, chlorophyll, to make food from sunlight. Vacuoles are storage spaces. Some store food, water, or waste. Golgi bodies and endoplasmic reticulum help package and transport materials inside the cell. 3. Cell membrane, a thin skin around the cell that controls what goes in and out, like food particles, water, waste, and oxygen. 4. Cell wall, only in some groups. Chrysophytes and some algae have a rigid cell wall made of silica, glass mass-like, or cellulose, a sugar-based material. Others, like euglena, have a flexible covering, pellicle, that lets them change shape. 5. Structures for movement. Flagella, long whip-like tails. 1 or 2 of these may protrude from the cell. Cilia, many short, hair-like projections that beat in rows to swim or sweep food. Pseudopodia, false feet, temporary bulges of the cell membrane that push outward to move or engulf food used by amoebae. Number 5. How protists make more protists. Reproduction. 1. Asexual reproduction, cloning. Binary fission. The cell simply splits into two identical daughter cells. Steps. 1. The cell copies its DNA, its instruction book. 2. The cell divides in half, giving each new cell a copy of the DNA. Budding. Less common in protists. A small bud grows on the parent cell, then breaks off to become a new cell. 2. Sexual reproduction. Mixing DNA. Cell fusion. Two protist cells come together and exchange or combine genetic material. They form a zygote, a single cell with mixed DNA. The zygote often becomes a resting spore or cyst, a tough shell, then later divides to form new protists when conditions are right. Advantages of sexual reproduction. Creates genetic variation. Kids are not identical to parents, so some offspring may survive better if the environment changes, temperature, food supply, etc. Number 6. Ecological roles of protists. 1. Primary producers. Photosynthetic protists, diatoms, dinoflagellates, chrysophytes, euglena in light, make oxygen and organic food. They form the base of the aquatic food chain. Tiny herbivores like zooplankton eat protists. Small fish eat zooplankton. Bigger fish eat small fish, and so on. 2. Consumers and predators. Protozoans, like amoeba, paramecium, eat bacteria and tiny particles in water or soil. Help control bacterial populations. 3. Decomposers and nutrient recyclers. Slime molds and some protozoans feed on decaying matter, breaking it down into nutrients that go back into soil or water. This recycling supports plant growth and the entire ecosystem. 4. Symbionts and parasites. Symbionts. Some protists live inside other organisms and help them, e.g. certain algae living in coral. Parasites. Some protists cause diseases in humans, animals, or plants, e.g. Plasmodium causes malaria, Trypanosoma causes sleeping sickness. Number 7. Why protists matter. 1. Oxygen and food webs. Photosynthetic protists produce a large portion of the world's oxygen, especially diatoms in oceans. They are food for many animals, from microscopic crustaceans to fish to whales. 2. Scientific importance. Protists, being eukaryotes, help us understand how more complex life, plants, animals, fungi, evolved from simpler ancestors. Studying them shows how a single cell can carry out all life's functions: growth, movement, feeding, and reproduction. 3. Human health. Some protozoan parasites cause serious diseases, e.g. malaria, amoebic dysentery. Understanding them helps in finding cures or prevention. Number 4. Environmental indicators. Changes in protist populations, such as algae blooms, can signal shifts in water quality, pollution levels, or climate changes. Number 8. Key takeaways. Recap in simple points. 1. Protists equals one-celled eukaryotic organisms, have a nucleus and organelles inside. 2. No clear borders. Some behave like plants, some like animals, some like fungi, and some do a mix. That is why Protista is a general, somewhat messy category. 3. 5 main groups in this book: chrysophytes, golden algae, diatoms, photosynthetic armored shells, live in water. Dinoflagellates, 2 flagella, photosynthetic or mixotrophic, can glow, cause red tides. Euglenoids, both plant-like, photosynthesis, and animal-like, feeding, flexible shape, 1 flagellum. Slime molds, look like slime, live on decaying matter, move as a big blob, form spores. Protozoans, animal-like, move by pseudopodia flagella cilia, eat bacteria or cause disease. 4. Mostly aquatic, live in ponds, lakes, oceans, damp soil. 5. Complex cell structure, have nucleus plus mitochondria, sometimes chloroplasts plus vacuoles. Some have cell walls, diatoms, others have flexible coverings, euglena. 6. Movement, flagella, long tail, cilia, short hairs, or pseudopodia, false feet. 7. Reproduxexual, split into 2 identical cells. Sexual, 2 cells join to form a zygote, mixing DNA for variety. 8. Big roles in nature. Make oxygen, form the base of the aquatic food chain, recycle nutrients, and some cause disease. Final thought. Protists are like the jack-of-all-trades of the one-celled world. They live mostly in water or moist places, and you can find some that look, eat, or behave like plants, some like animals, and some like fungi. Their cell is well organized. more complex than bacteria, but simpler than a plant or animal body. By studying Protista, we see how life formed basic building blocks that eventually led to all the complex plants, animals, and fungi around us today. Remember, Protista is not a perfect, neat box. Scientists still talk about who belongs and who doesn't. But knowing about protists helps us understand ecosystems, human health, and the evolution of life on Earth. I hope this deep, detailed summary helps you see every important piece of what Protista is and why it's so fascinating.